All right, this video is going to be sort of a mixture of various resources that I've got for the digestive system. It is important for class, however, because there's going to be some stuff to draw and some notes to take, and you'll be finishing up the digestive anatomy lab and submitting that at the end of this. So the first thing that we're going to look at, however, is the small intestine, pancreas, liver, and gallbladder. You guys will have a printout for that from my website, and we will have already done some of that. So if you could find that real quick, and as you're looking for that, I'm just going to show you that I've got the information right here for that, all right? And I'm going to start filling in some of the blanks. So just as you're looking for that, just note that we've done some of this stuff already. However, just recall that I've made some changes to this. I've kind of decided that we wouldn't do anything with the term prosecretin. I'm going to focus on secretin, remembering that secretin is a hormone, all right, that's going to be released from the small intestine when acid enters the small intestine from the stomach. That's H+. Acid is H+, all right? So when the H+ goes into the small intestine is going to release the hormone secretin. That's going to travel through the bloodstream, all right? It's going to go down to the pancreas, and it's going to cause the pancreas to release something called bicarbonate, HCO3-. The bicarbonate is going to go into the small intestine to neutralize that acid. Now, let me see if I've got anything important here. Now, I've got an overview of the digestive system here. I guess the pancreas is sort of underneath there, number 5. This doesn't really help. So if I come over to this one, it's not going to stay. So I got to mouse over that in order to do it. But you can see the pancreas over there on the right-hand side, and you can see the pancreatic duct. And if you can notice, there's a spot where it says common bile duct opening. You'll see. Let me see if I can just mouse over this and have it stay there. If I do that, yeah, that doesn't quite work. So I'll try it this way. But if you can, okay, sorry, I'm probably going to edit that out, but if I don't, it'll be funny. Anyways, you can see the pancreas there, and you can see the pancreatic duct. This is where the bicarbonate and the various enzymes are going to travel into the small intestine to do their work. Okay, so in this case, bicarbonate. Now, I do have an animation that I'm going to want to show you. This is a very old Flash thing that I made years ago, and I don't really like the way I did this. But anyways, the bicarbonate is going to neutralize the acid, and that's going to inactivate pepsin and provide the conditions for trypsin to work. So let me see if I can find that little animation. I've got this on my website as well, and we'll see if that can help you to visualize what's going on. So we want the secretin one here. So again, yeah, you could search for this on my website. It says this animation illustrates the interplay between secretin and bicarbonate in the neutralization of stomach acid. All right, so we're going to do this. When food enters the stomach, glands in the stomach wall release hydrochloric acid, HCl. The acid is needed to activate enzymes that digest proteins. But when the partially digested food along with the acid moves into the duodenum, the acid must be neutralized. All right, so let's have a look at the organs of the digestive system that are involved in acid release and neutralization. So basically what you do here is you just mouse over each of these, and it tells you what they do. You know, the esophagus transports the food in. The pancreas is going to produce that bicarbonate. And there's the pancreatic duct, which will transport the bicarbonate from the pancreas here into the duodenum, which is where the acid is going to be neutralized. So here's what happens. All right, let's cut away. You can see the gastric gland. So let's watch this process. There's the protein coming in. Now, the way I've drawn this is the acid cleaves the molecule here so that pepsin results. But back here, I had just pepsin. But really, the acid converts pepsinogen into pepsin. And as you can see, pepsin broke the protein into smaller fragments, not right to amino acids, as smaller peptides or polypeptides. That acid is then going to travel down, so let's check that out. We don't need to read that stuff. The protein and the acid and all the other stuff is going to go down into the duodenum. So there it is in the duodenum. So now this acid needs to be neutralized. So we will see the release of secretin. So there it is, goes to the pancreas. So the secretin came from here, down, and it goes over to the pancreas. The pancreas will then release bicarbonate, which will neutralize the acid. All right, and we're done. So you can see just a summary diagram, animation of the whole thing. There's the protein, there's the pepsinogen, really. Acid cleaving it to pepsin, pepsin breaking the protein into fragments. All of that going into the duodenum. Secretin being released from the wall to the pancreas to release bicarbonate to neutralize the acid. And of course, there's a complicated chemical reaction that you need to know about that. So that's all I wanted to show you about that part, the secretin. And then we moved on and we mentioned that the pancreas also releases various enzymes. We've got this trypsinogen here. The trypsinogen is converted into trypsin by an enzyme called enterokinase, which is in the small intestine. Now, I don't have an animation for that. Trust me, it happens, though. All right, and trypsin's going to act on partially digested proteins or regular proteins, and it's going to break them down into peptides. So again, we're still at the level of chains of amino acids. We're not at the amino acids yet, but then we get another one called erepsins, which are also from the pancreas, and I believe they're also from the small intestine as well. And that finally converts them down to amino acids. So we haven't yet seen how the amino acids or small carbohydrates are absorbed into the bloodstream yet. We're going to see that today. That's one of the things that you're going to be doing. So we have that, and we have this. Whoops, sorry about that. I got to curse down all the way. How about that, eh? The pancreas also releases amylase. So there's salivary amylase breaking down starch to maltose and pancreatic amylase breaking down starch to maltose. All right, so here we have maltose, a disaccharide, and then there are various enzymes in the small intestine that do chemical digestion. These are called disaccharidases. Enzymes that break down disaccharides are called disaccaridases. All right, for example, maltase or lactase or sucrase. And they're going to break them down into monosaccharides, for example, glucose, in the case of maltose. So maltase will break down maltose into glucose. All right, so I believe we already have this stuff, and we may have even more. And then there's lipases that break down lipids into glycerol and fatty acids. And again, there's a gastric lipase, and this is a pancreatic lipase. So there's a lot of enzymes flying around here, right? Now, the liver and the gallbladder. I was trying to show you some images earlier. There you can see the liver and you can see the gallbladder underneath that. You can see the cystic duct coming right off of it, and then you can see the bile duct, which comes down and joins the pancreatic duct, bringing both bile and pancreatic secretions into the duodenum. Okay? So, yeah, this is a really weird way of doing it. The liver produces a fluid called bile, right, which contains bile salts, which speed up lipid digestion. Now, this is not really important in bile salts, but the liver makes bile, which helps to speed up lipid digestion. Bile is stored and concentrated in the gallbladder. When there are fats in the small intestine, we have another hormone, and this hormone is called CCK, which stands for cholecystokinin. All right? When the fats are in the small intestine, CCK is released. It's carried by the blood because, remember, CCK is a hormone. You need to have your hormones straight from your enzymes. Remember, enzymes are the workers, hormones are the messengers. They tell workers what to do, all right, kind of like teachers. All right, so CCK comes from the intestine and it goes to the gallbladder, and it tells the gallbladder to release the bile. The bile then goes into the small intestine. Remember, it doesn't go into the stomach, it goes in the small intestine. And this is where the bile salts, or the bile, emulsifies the large fat globules into smaller droplets that allow lipase to work. Now, I'm going to show you an animation for that. All right, here we go. Over here, same idea. So I'm just going to go quickly through this. You can see this on my website. You can read about that. But let's cut away and let's see what happens when a cluster of lipids come in. There's a big chunk of fat into the stomach. Now, gastric lipase tries to break that down, and it might actually get some of the external triglycerides or fats there, breaking them into glycerol and 3 fatty acids, but it can't do much about it. All right, but we start fat digestion there. So the lipid cluster then goes down into the duodenum along with all the gastric contents. And there it's going to trigger the release of a hormone. Now, we saw secretin earlier going to the pancreas. This time, it's going to be CCK going up to the gallbladder. So you see CCK came out of there and it went up to the gallbladder. That CCK hormone is going to tell the gallbladder to release bile, which breaks down the large lipid into smaller pieces, all right? It emulsifies it, breaks it apart, all right? And now an enzyme from the pancreas, lipase from the pancreas, is able to break down those individual lipids into glycerol and fatty acids. But now, remember, you have the glycerol and the fatty acids. We have to see how those are absorbed into the bloodstream. So that's what we're going to do soon, right? So we have to account for all of that. All right, so that's how that process works if you want to see the whole thing. Large lipid cluster, gastric lipase does what it can. But if there's a big chunk of lipid still into the duodenum, release CCK, the hormone, through the blood to the gallbladder to release bile to emulsify the lipid, so pancreatic lipase can now break it down to glycerol and 3 fatty acids. Okay, so it's a complicated business, admittedly, with those hormones. You had secretin, you had CCK, and of course there are others. Now, for this last couple of parts, I don't really want to say too much about this. It just mentions cholesterol here, just to show you this is the structure of cholesterol. All right, we'll study that in grade 12 bio a little bit. Can this go backwards, actually? No, apparently not. Okay, we'll do that. So cholesterol is part of bile. You need cholesterol in your diet. You always hear this bad stuff about cholesterol, but cholesterol is actually a part of your cell membranes. All right, study that in grade 12 bio as well, how it does that. But it's a necessary part of your cell membranes. You might remember that it creates steroid hormones. We studied some of those steroid hormones when we did sex-linked disorders. It's the building block of testosterone and estrogen and progesterone. So you need cholesterol, and it's also part of your bile. But if you have too much, then the bile starts to precipitate or come out of solution, sort of like salt at the bottom of a glass of salt water. You know, you get this crystallized type stuff, and that can form gallstones. And the gallstones can block the bile duct, very painful, and impairs the release of bile, which is necessary for fat digestion. All right. So, I mean, the gallbladder can be removed. You can live without a gallbladder. Your liver can still produce the bile and release it through the duct. It's just that, you know, you don't have the storage of the bile. So you have to watch what you eat. You have to go easy on the fats. I'm not really going to worry about this other stuff here. All right, so the important thing is just that cholesterol is part of bile. And that's really all there is to it there. However, this last part is important to me, and I'll say more about this in class. The liver, in addition to creating bile and storing it and needing cholesterol, it produces some vitamins. All right, I'm not going to worry about which vitamins. Just know the liver produces some vitamins. That's necessary. The liver detoxifies the blood, and when I say that, I don't mean filters, all right? The kidneys filter blood and produce urine, liquid waste. The liver detoxifies the blood, so all of the blood goes through the liver, and all of your food and all of the drugs that you might have taken, allergy medicine, headache medicine, anything, all right? And any other bad stuff, you know, food coloring, bacteria, alcohol, whatever it is, the liver has to deal with trying to convert it to a less toxic substance and then making it water soluble so that you can urinate it out or something like that. So the liver is under a heavy load of doing that, but it does a good job of doing that. So that's detoxification. That's not really exactly a digestive function, except for the fact that whatever you ingest through the digestive system will eventually make its way through the liver, and the liver will have to deal with it. But the other thing that I want to talk about, and you know how much I like my biochemistry and the hormones and the enzymes, glycogen is a polysaccharide, and we tend to draw our polysaccharides like this, right? Long chains of glucose. All right, that's actually what glycogen is, long chain of glucose. Well, what's the other one you've seen? Well, you've seen starch as a polysaccharide of glucose as well. Starch is in plants, glycogen is in us. So when you eat your meal and you get a bunch of glucoses in there, you're going to use those glucoses for energy, all right? But you have more than you need, and the body needs to store it up. So for those times that you do need energy and you're not eating at that moment, what happens is that glucose goes into the liver, and it is put into a chain. It is polymerized into a polysaccharide called glycogen. Now, glycogen actually has branches of glucose like this. All right, the glucoses are all over the place, like, something like that. Whereas starch is just a more linear chain. Glycogen is more branched. It's not an important point for grade 11. It will be for grade 12. But this is a storage form for glucose. Just like when you wash your knives and forks and spoons, whatever else you have for your utensils at home, right? You probably don't just throw them into the drawer all over the place. You might, if you're a bachelor or bachelorette. But chances are you put them into the little plastic thing, with the spot, it fits the shape of the fork and the spoon. So it's all nicely arranged and it takes up less space. It's the same thing with glycogen. It packages the glucose together. And when you need more glucose, your body can cut the glycogen, releasing the glucose. Now, that, of course, will take an enzyme. We're not studying that, and I'm not going to cut every single one. But the glucose can then be released into the blood. There is a hormone that causes the glucose to be stored as glycogen, and it's called insulin. And we'll study that a little bit later on, all right? Insulin lowers blood sugar by putting it into the liver and storing it up as glycogen. Insulin causes glucose to go into the liver. Another hormone, which is called glucagon, not glycogen, not glucose, glucagon, and you'll look at the written form of that word later. Glucagon causes the glucose to come out of the liver into the blood. So if you need energy, you go, hey, I've got all this stored glucose. Why don't you chop it up and release it? Well, that's what glucagon does. It goes to the liver. Actually, both of these hormones come from the pancreas through the blood to the liver. And glucagon says, hey, that glycogen you got? Well, we need some glucose. Chop it up and release it. See the way the hormone is the messenger? Glucagon doesn't go and physically cut it. It tells another enzyme, cut it and release the glucose. So I do want you to know about glycogen, that it is a polysaccharide like starch, and it's formed of glucose. And we're not learning the enzyme like we learned amylase to break this down. We're not learning that right now, okay? That's basically what I want you to know from this stuff here. Okay, is this, I got an interesting picture here at all? This is just showing cirrhosis of the liver. When the liver gets damaged, too much toxins, too much alcohol, it can die. And you need your liver to detoxify stuff, so you have to take care of it. Okay, so I wanted to show you that. I also wanted to show you a couple of other things. Let's have a look at Dinis Diner. I named this after my wife, Dini. This is a little game that I made up for the digestive system. All right, you're sitting at your table here, ordering your meal, and on sale, pickled pigs, pylorus, whatever, don't ask. So we're going to order some food. I'll be your waitress tonight. Here's your menu. Okay, so you now click on the menu. This is on my website, of course, right? And you say, okay, so what do I want to order? And you read a question. The intestine uses this structure for absorption, and here's how much it costs. So which one does the intestine use? Well, we're going to talk about that later. Let's assume we think it's ruga, which is in the stomach. Let's order some ruga, because that answers the question. You go like that, and they serve you your meal. Okay, let's have a look at the meal. Oh, that's terrible. Look at the flies going around in that. Okay, so that's no good. All right, let's try another one. Oh, no happy customers, because that was a bad meal. So we go to the next one, main meal number 2. The taste buds are often found on the sides of the, okay, choose the right menu item for this one. Oh, they accept tips. I'll show you what that means. Papillae, right? Okay, papillae. That's what I want. I'm going to order, eat some papillae. Let's check it out. Oh, that looks good. Okay, so what you do now when you get a nice meal is you click on it, you eat it, and there's some money. All right, because you ate it, you gave some money. So you click that, and that goes to Dini. And some fries. Click that, that goes to Dini. It says how much tips you've given. And if you give full tips the whole game through, you win a prize. Oh, Dini's needed in the kitchen. So then we go over to the kitchen. You can see a little stove here. And this is like the menu rack, right? So I don't know if you can read this. Most absorption of nutrients occurs in that, and you got to choose one. Let's choose the mouth, okay? And then check the stove burner. I'm going to choose D, and let's look at the burner. No, that didn't work. That's crossed off. I'll choose the right one and see if the burner works well. See if I know how to cook here. Choose the right. Oh, look, it heats up. That's the right one, okay? And basically, you'd go through all this and you'd go back to the restaurant and you play the game. I just want to show you that. I'll show you one more quick one before we do some work. I can't even remember what this one is called, but flip the power switch to load the drinks. It's a drink machine. So there's the power switch. Ching. And there's the drinks. Oh, and we got a stomach there. Get a coin and insert it into the drink machine. So it says click for coin. This one's also on the website. Okay, there's a coin. What do? You click and drag. Oh, you click and drag. I'm using the tablet pen. Bring it up to here and let go, and it just goes automatically in. So now the questions are available. What is the first tissue layer of the esophagus called? Anybody yell it out? See? Mucosa. Okay, C. So let's see if this works. See? You get a drink. Take a drink, and your stomach is satisfied. And you got to get another coin. Bring it up. Put it in. Question 2. What enzyme is present in saliva to break down starch? Let's try the wrong one, see what happens. B. Just says no. Amylase. Ding, there's a drink coming down. Get a drink. Your stomach's... Okay, that's how it goes. Okay, vending machine, that's what it's called. Okay, obviously, right? I just want to show you this quick one. This is a really old little thing just to show you a potential gastric gland could work. If this is the gastric gland here and this is the lumen of the stomach, you've got a protein made of a bunch of amino acids. If you click on one of these cells, you can see it releases HCl. And then pepsinogen, the HCl converts it to pepsin, which breaks the protein into peptides. And that's all there is to that. Good times, right? Restart, you can watch it again. Okay, now on with the main business. I'm not going to do that one today. So what I want to do is a little bit of this. Okay. Could you please find your diagram of the anatomy diagram I gave you at the beginning of the course? And when I say course, I mean strand. And when I say strand, I mean digestive system. You should already have all these labels. Actually, let me do this. Okay. You should have all of these labels already done. We've seen the stomach here with the gastric pit and the gastric gland, the various layers of muscle, and et cetera, et cetera. So it's not, I don't want to focus on this right now. I'm going to hope that you've already seen that. What I do want to focus on is this diagram right here, okay? This is the villus. So we're moving through the digestive system down into the small intestine. You're going to be drawing this very soon. I want to do something to do with the villus. So, of course, the villus has these projections. This is a villus. There's another one, etc. These are called villi, or one is called a villus. And we have the same layers. So the villi are really projections of the mucosa layer. We say the mucosa is thrown into finger-like projections called villi. For the stomach, we say the mucosa is thrown into folds called rugae. In the intestine, we say the mucosa is thrown into finger-like projections called villi. So we have the mucosa, the submucosa, and then for the muscularis, just the two muscle layers, circular and longitudinal muscle, as opposed to having the oblique. But now, if we zoom in on one of these villi, and I know you already have these labels from before, I want to talk to you about the absorption of nutrients. Things like, and this I want you to add to your diagram, things like glucose, when we get all the way down to the monosaccharide level, whether it's glucose, and there are other monosaccharides, there's fructose and there's galactose, all of these monosaccharides, and you can use glucose as your main one for now, and amino acids, all right, from the protein breakdown. These things are absorbed through the villus into the capillary system. All right, these are the capillaries. All of these are capillaries here. There's an arteriole bringing blood in. There's a venule bringing blood out, but the smallest diameter blood vessels are the capillaries, and the capillaries is where these final nutrients are absorbed from the digestive tract into the bloodstream. And now the bloodstream will deliver them to wherever they're needed. All right, now there are other tiny molecules that can be absorbed in the capillaries, but we're focusing on these are, remember, these are from our carbohydrates, these are from our proteins, all right, so those major groups. The other group we have are the lipids, and the lipids, when they're broken down, produce fatty acids and glycerol. Now, I may not be drawing these in the best spot, and I don't like that at all. Let me do that again. Fatty acids and glycerol. These are the breakdown products of triglycerides, or we'll call them fats. They are absorbed into the lacteal. That's that central worm-like structure right in the middle. Okay? So fatty acids and glycerol are absorbed into the lacteal of the villus. This will eventually join up with the regular circulatory system. There's a slightly different circulatory system called the lymphatic system, and it carries lymph instead of blood. It's kind of a yellowish, greeny kind of fluid, all right, like plasma kind of, all right. So we have absorption of some nutrients, and these ones, of course, come from the lipid category. All right, so those are our three major macronutrients, right? The lipids, the breakdown product being fatty acids and glycerol, into the lacteal and eventually into circulation. And on the other side, for the carbohydrate family, right, macronutrient, you may not know macro versus micronutrients yet, you will soon. Glucose as our typical one. And from the proteins, amino acids, they go into the capillaries. This is absorption of nutrients. All right, so imagine a question on a test or exam talking about the breakdown of protein. Let's take a protein and, break it down completely all the way to the level of absorption. So you'd say, okay, in the oral cavity, there's nothing going on. Down the esophagus, nothing going on. Into the stomach, yes. This is where pepsinogen is converted into pepsin using HCl, right? Pepsin breaks the protein into peptides or small chains, okay? Into the small intestine. This is where trypsinogen from the pancreas is converted into trypsin in the small intestine by enterokinase. And we get peptides again for any proteins that weren't broken down by pepsin. All right. You also have in the small intestine erepsins, which break those peptides finally down, finally down into the amino acid building blocks, which are now absorbed into the capillaries of the villus and transported. So see how long that process is? And imagine along the way needing the correct pH of, 2 in the stomach and 9 in the small intestine and all that, and the correct temperature, or else denaturation occurs. So, imagine having to know the entire protein pathway and the lipid pathway and the carbohydrate pathway, and that's what you will have to know. All right, where am I going now? Okay, I'm. You should have all those labels on your diagram. Now what I'm going to do is get out of there. I'm coming over to something else that I want to show you, which is this digestive anatomy lab. Just a little quick review of this. Now, I'm just going to do something here for a second because my camera shuts off at 30 minutes. So give me a second here. All right, I have to shut the camera off at 30. I could probably find another setting for that, but okay, we're good to go. All right, just check this out. This is going to lead to the final bit of the class. Just really quickly, as a bit of a review, you've already seen this in the lab, right? The papilla, which is really this whole region on the surface of the tongue. There are thousands of these papillae, all right? And along the lateral borders here are the taste buds, all right? And these taste buds are really collections of taste cells. There are many cells in here making up a taste bud. And there's a tiny little pore here, so the food that you eat and chew and that saliva lubricates, the food comes down and it stimulates a little hair here, a little nerve, and that sends a nerve impulse up to the brain. Okay, so those are the taste buds on the papillae of the tongue, which you've already seen. Here's a blow up of those. Again, you can see that if this is a taste bud, I don't know if you can see, that's a nucleus and another nucleus, etc. So these are all the taste cells, and they even show a little bit of a cranial nerve here, so the nerve impulse would travel down this nerve and go off to the brain, and you'll study nerve impulses in the grade 12 biology. Okay, you've already seen the cross-section of the esophagus with the lumen here, the mucosa, the submucosa, and I know it's difficult to tell what everything is. Just, what you got to do, really, extend that to there, is find your landmark features. If you start at the lumen, you can easily identify the mucosa. If you start at the outer edge, you can easily identify longitudinal muscle. Often circular muscle, you will see the sort of the gradations going around, okay, the pathway, and so you can identify circular. So you can distinguish between longitudinal and circular and mucosa. So then everything in between, you're going to call submucosa. All right. So that's the esophagus we've already seen. I thought this was an interesting image to show you as well. This shows where the esophagus and the stomach join. All right. You might recognize the nice smooth layer of the mucosa of the esophagus coming down to what looks like octopus tentacles. These are all gastric glands. All right. So this is the stomach and this is the esophagus here as they join. And of course, the cross-section diagram or some microscope slide of the stomach itself, again with the lumen. And then we've got the mucosa here with a lot of gastric glands. Some of those will be down here, the mucosa, and then it gets, or the submucosa, and then it gets very complicated for the muscularis with the 3 layers. So we're not going to worry about identifying them, but you could almost see longitudinal, circular, and you can see some stuff going like this in weird angles. You might be able to catch the oblique there as well. All right, so there's the stomach with gastric glands, etc. And I'm zooming in here a little bit on gastric glands. So each one of there's aruga. And these gastric glands, I don't know if you can get a sense, but they come down here and they kind of open up between the rugae and release their contents. These ones are, it's hard to tell that that's a gastric gland, but I mean, there could be a tube coming up in behind that you just don't see in the way that it's cut. But anyways, those are the gastric glands releasing gastric juice. And this is really where we leave off here and where I'm going to end the video. What I'll ask you to do is to pause the video at this point. And this is what I'm going to have you draw. But before you start that, just hang on a second. All right. This is the small intestine. All right. You're looking at the lumen up here. And then the wall of the intestine, this is outside of the intestine, and this is inside of the intestine. You've got a big layer, which is mucosa, thrown into those finger-like projections. Each one of these is a villus. Now, you might look at this and go, well, what the heck's that? But remember, these are like trees, all right? And you know the way a tree isn't straight? It kind of curves like this, all right? So when the slide is cut, some of these break apart. You only get fragments. But these are the villi, the structures that actually absorb the nutrients. And so if you had amino acids in the lumen, they would go into the villus. And if you had the right microscope image, you could see the capillaries and the lacteal in the middle for the fat absorption, the breakdown products of fat. So this is the mucosa. But there are also intestinal glands here that release all those enzymes, the enterokinase, the maltase, the sucrase, etc. So all of these circles are intestinal glands releasing their contents into the lumen. There is a small submucosa here, and then there's circular muscle, and you just got to try to imagine that it goes around like that, and then a layer of longitudinal muscle that would be coming sort of out at you here as well. So you have both circular and longitudinal making up the muscularis in total. These are the labels that I want you to draw for this. Hopefully you can find some blank white paper. You're welcome to draw it on the back of another image. All right. But scientific or biological format with your name at the top right and all these to the right side from shading and stippling. I figure it's going to take you 15, 20 minutes. You should be able to get that done. When I see you, I will show you the real thing. I only have a couple slides of this, so not enough for all of you. So I want you to see the real thing, but for now, you're seeing a picture of the real thing. Then I want you to see the microscope slide of the real thing. Small intestine is the name. Draw this nicely, okay? Spend the rest of the time doing that if that's what you need to do. And once you're done, you are going to submit your entire lab, okay? Make sure your name is on every page, and you should have the teeth, the tongue, the esophagus, the stomach, and the intestine. Now, if you've been away for any class, I want you to leave me a note to remind me about what image you did not draw. All right? And then I'm not going to mark that for you. I'll omit that. But you need to tell me what's going on. Otherwise, I'm expecting all of the drawings. All right? If you could try to put them in this order, that would be very helpful. And try to have them, right side up, nice way, so it's a nice little package that you're proud of. That you're going to staple it together, submit it, all right, and it's done. And I'll take it from there. If you have the lab printout that tells you all the things that you needed to do. If you could staple that on the front, that would also be good, but it's not absolutely necessary. All right, so spend the rest of your time working on that, unless I come up with something else that I need you to do. Just do that. So pause the video here. I'm going to say goodbye, and good luck drawing that, and thank you for watching.